Now that we've discussed and learned how DNA actually replicates, we want to turn our attention to how the genes within DNA sequence are used to actually produce polypeptides and proteins. And this process is called or referred to as protein synthesis and includes two primary stages of transcription and translation. And so this video, we'll look at the transcription process, and this is for D1.2. Transcription is the synthesis of RNA using DNA as a template. The RNA that is created or copied, or you could call it transcribed, is a single strand that, and it only occurs along one strand of the DNA. So DNA obviously is a double helix. There's two strands. RNA or messenger RNA that is produced through the process of transcription is going to be single strand. This can occur repeatedly as needed so that DNA can be copied into this messenger RNA numerous times, as much as necessary, and the process stops when it gets to the end of the gene. The copied strand, the messenger RNA, is identical to the DNA strand that's opposite of it. So we refer to the two strands of DNA, the sense strand and the antisense strand. And that messenger RNA that gets produced is identical to the DNA sense strand, except for that uracil replaces thymine. The enzyme that actually conducts this process is called RNA polymerase, and it binds to the DNA at the start of the gene. It unwinds the DNA and then separates it into two separate strands. And it moves along the template and positions RNA nucleotides on the growing RNA strand so that they're complementary to those on the DNA template. So if we think of this as similar to DNA replication, it's kind of the process of helicase and DNA polymerase 3 together in a single enzyme, and that's RNA polymerase. It also then links the nucleotides by covalent bonds between the sugar and the phosphates. Between those two, creating covalent bonds, and then it detaches from the DNA, allowing the DNA double helix to reform. So this process doesn't damage the DNA, it doesn't destroy the DNA. As soon as it's done, the DNA winds up again and is ready to be able to be used again in the future. The product then is what we call messenger RNA that will eventually leave the nucleus, at least in eukaryotes that do have a nucleus, and it joins a ribosome in the cytoplasm for the second stage of protein synthesis, the steps of translation that we'll look at in our second video. Just as in the DNA molecule, hydrogen bonding is also important and essential to the replication or the production of this RNA strand. Nucleotide bases are complementary because they form specific hydrogen bonds with one another and not other bases. And so cytosine and guanine bind together, and adenine and thymine bind together. Guanine and cytosine have 3 hydrogen bonds, adenine and thymine have 2 hydrogen bonds. And so the DNA strand that's used as a template, as I mentioned, is called the antisense strand, meaning that the sequence of the RNA that's produced is the same as what's on the DNA sense strand, the opposite side, except for the replacement of the uracil and thymines. And that's again dependent on the complementary pairing of those hydrogen bonds, where uracil will also have 2 hydrogen bonds, as does thymine. The DNA as a molecule is really stable, and this is helped or supported by the fact that it's kept within the nucleus. Really, the DNA sequence needs to be maintained as much as possible for the survival of the individual and the species. The splitting of the DNA molecule for the production of RNA does not cause damage or cause changes to the DNA sequence. After transcription, the two strands are linked back together and linked by hydrogen bonds, as we just discussed. So they're really only parted or separated for a short period of time by that RNA polymerase enzyme, and so they're really only briefly vulnerable to chemical changes that might change the sequence. And so the stability of the DNA is essential to the survival of the cell and that organism and the species, because that gene will be transcribed many times during the cell's lifetime. And if mutations were to occur, they would quickly become disastrous, as they would increase in the number if the DNA was not stable during this entire process. So there'd be more and more mutations that would develop, and that would probably lead to the destruction of the cell and potentially the organism, and would have consequences for the species overall. So it's really important or essential that the DNA is stable and does not change during this process. The expression of genes, or gene expression, is taking the information within a gene and using it to be able to produce proteins. And that protein then has an effect on organism. The gene specifies the sequence of amino acids that will be in a specific polypeptide. That polypeptide then will be folded into a particular protein, and that sequence of amino acids, as it's called for by the gene, we refer to as the genotype. It's literally the sequence of nucleotides that are found within that specific gene. The protein that's produced then either directly or indirectly determines the observable characteristics within that organism, or sometimes non-observable characteristics, but it's responsible for the characteristics of that organism, and we refer to that as the phenotype. And so, as an example, humans have genes that are responsible for the production of specific hair color. That information comes from the genes. The phenotype then would be the actual hair color that we see. This is dependent on these two steps of protein synthesis, transcription and translation. Transcription is the first step in which the messenger RNA is produced, as we've discussed. It's not always active. It can be turned on or off. Genes can be switched on or off at any given time, and some are never activated during the life cycle of the cell. And different types of cells have different genes activated. So the full range of RNA types made within a cell is called the transcriptome. And again, different cells can have different types of genes that are turned on or off at different points in times. And that's really what is responsible for producing different types of cells, is the activation of some genes versus others in different types of cells. Patterns of gene expression can vary by the level or the amount of the gene that's expressed. And it can allow for cells to differentiate for different types of functions. Within our image here, we see the expression of patterns of a fruit fly with expression of different genes in different locations during the different times of development. And so genes can be turned on or off or expressed at different times throughout the organism's life cycle, as we've discussed, or they can also be expressed in different amounts. So there could be higher amounts of one gene versus less amounts of a different gene, and that can then have impacts on the development of that organism or that cell. The gene expression is regulated by transcription. So genes are not always activated, and they won't always be transcribed, even though they persist within the cell's DNA, the DNA that's used to make the messenger RNA. So gene expression is not always continually producing proteins. It's dependent on the needs of the cell or the development of the cell and that organism. A promoter is a section of DNA that initiates gene transcription. It's usually 100 to 1,000 bases, and it's located next to the gene on that chromosome, so the same chromosome, and it allows RNA polymerase to bind with the transcription factors, different proteins that are attached, and there's different types of transcription factors. There can be repressor sequences within the promoter region that also allow the blocking of transcription factors from being able to bind to the gene, and so then that would block the binding of the RNA polymerase and thus transcription overall. There can also be activator sequences within the promoter area that are essentially the opposite and allow the binding of transcription factors and thus initiating RNA polymerase and leading to transcription. Gene expression can change depending on what transcription factors are bound within the promoters. They can be a single molecule, those promoters. They can be a combination of multiple molecules. It really depends on the gene in terms of what's activating or repressing and what that molecule, what those proteins are that activate or repress that process. Of all of the DNA that we have within our cells and other organisms have within their cells, the vast majority is not used to code for polypeptides and to produce proteins, but they actually have other functions. And those can include being transcribed to make tRNA, transfer RNA that we'll talk about a little bit more later, or rRNA, which is used to produce ribosomes. They can also be used in gene regulation and expression, such as promoters, enhancers, or silencers, repressors, as we just discussed. They can also be found in telomeres, which are the end caps of chromosomes, and some research suggests that maybe that these have some connection to the aging of cells. And they can also represent introns, which are base sequences that are removed from messenger RNA during post-transcription modification, specifically within eukaryotes. And we'll talk about a little bit more here momentarily. After transcription is complete, what happens to that messenger RNA is different within prokaryotes and eukaryotes. In prokaryotes, that mRNA is translated immediately after transcription, so it goes directly to the next step, to produce that protein. In eukaryotes, the messenger RNA is altered into a mature form of messenger RNA before it leaves the nucleus. And this is one of the reasons why the nuclear membrane is necessary to be able to contain that RNA before it can be released. It needs to be modified to form mature RNA. And it also allows, the nuclear membrane also allows for the separation of transcription and translation to allow these modifications to occur. So within eukaryotes, the RNA modifications that take place, the first would be a 5' cap. And so this is a modified nucleotide that's added to the 5' end of the RNA that has 3 phosphate groups instead of only 1. So it's somewhat similar to ATP. Additionally, a poly A tail is added to the opposite end, the other end, and it's usually between 100 and 200 adenine nucleotides that are added at the 3' end of that molecule. And both of these, the 5' cap and the poly A tail, help to stabilize and protect the ends of the messenger RNA from being digested by nuclease enzymes. Additionally, there needs to be the removal of the introns that we just discussed. Eukaryotic genes have alternating sections called introns and exons. Only the exons are used in mature RNA. The introns, which are usually about 20 to 200 nucleotides long, must be removed, and they are digested into single nucleotides, allowing the remaining exons to be connected together, stitched together, and that's what forms up the messenger RNA. The removal of these introns is carried out by SNRPs, SNRNPS, and they are RNA protein complexes that recognize the sequence in the introns in order for them to be able to be removed. And introns make up a portion of this DNA, as we discussed, that doesn't directly code for proteins. To conclude the discussion of transcription and the process of producing this messenger RNA before it's used in translation, one last modification or process that can change the messenger RNA that's produced is alternative splicing. And this is a change that allows several variants of a polypeptide to be produced from one single gene. So essentially we can get lots of different proteins produced from the same gene. The initial transcription process and product is the same, but it's occurring post-transcriptional modifications that then alter the final product. Really, ultimately, it's oftentimes exons being skipped or not included, kind of like the introns. And it allows the change to form and function of the polypeptide without having duplicate genes, so it cuts down on the number of genes that are necessary. So a good example that we can look at is tryptomyosin protein that's found within mammals. The gene itself has 11 exons, and different types of tissues have different forms of the protein. Skeletal muscle, for example, lacks exon number 2, whereas smooth muscle, has is lacking exons 3 and 10. And so by these two having or lacking different exons, it changes the type of cell that they are. And ultimately, alternative splicing increases the diversity of the proteome, which is all of the proteins that are expressed by a cell or an organism. And so that concludes our discussion of transcription and how, specifically in eukaryotes, mature messenger RNA is produced. That will be used in our next step, which we'll focus on in the next video, the process of translation, using that to be able to actually produce a polypeptide and a protein.